Let's talk about what might just be humanity's longest-running, highest-stakes science experiment ever, the evolution of our food. I mean, think about it. Every time you take a bite of a Honeycrisp apple, or a slice of bread, or even just a handful of rice, you're literally tasting the results of thousands of years of crop improvement. In this explainer, we're going to unpack exactly how science has shaped our fields, and more importantly, where it's taking our plates in the future.
Picture the produce section at your local grocery store. You've got those massive, perfectly uniform ears of sweet corn, plump tomatoes, drought-resistant wheat making up your favorite pasta. None of that is just a happy accident of nature. They are highly engineered marvels. But we've got a massive challenge staring us down right now. With the global population booming and our climate changing faster than ever, these plants have to evolve fast just to survive. So, the big question is, how do we actually engineer the food of tomorrow?
Section 1. The Quest for Better Crops.
Okay, let's get our bearings and define exactly what we mean when we talk about this quest. At its core, crop improvement is just the process of modifying the genetic characteristics of crop plants. But, we're not just making changes for the sake of it. We're on the hunt for superior varieties with really specific, highly desirable traits. We're talking increased yield so we can actually feed a growing world, disease resistance so an entire harvest doesn't just get wiped out overnight, and, crucially, stress tolerance, so these plants can handle whatever crazy weather the environment throws at them. This right here, this is the scientific baseline for everything we're about to explore.
And what's really fascinating is how these objectives map so perfectly onto our biggest global challenges. Like, we aren't just trying to grow giant prize-winning pumpkins here. These are actual solutions to global food security. By improving nutritional quality, we're directly fighting malnutrition. Developing disease and pest resistance, that prevents devastating famines. Enhancing drought and salt tolerance means we can actually grow food in regions that people used to think were completely barren. And if we can reduce our reliance on chemical inputs like heavy fertilizers and pesticides, we're protecting our ecosystems while building truly climate-resilient crops. It's a win-win.
Section 2. The Traditional Methods.
So, how did we actually start doing this? Well, long before anyone had shiny laboratories or DNA sequencers, our ancestors were already out there hacking plant genetics. Let's look at how these traditional methods evolved. It all kicked off with step 1, selection. Really straightforward. Farmers just picked the absolute best, most robust plants from their fields and saved those seeds for the next year. Simple, right? Then came step 2, hybridization. This is where things get interesting. People started intentionally crossing genetically different plants, maybe different varieties, sometimes entirely different species, just to combine their best traits and create what's called hybrid vigor. But eventually we kind of hit a wall. We needed more genetic variation than nature was giving us. Enter step 3, mutation breeding. This is where scientists intentionally use things like X-rays or chemical mutagens to literally force new genetic variations. And finally, step 4, polyploid breeding. This is a wild one. It's a technique where you actually increase the number of chromosome sets inside the plant to fundamentally change its physical traits.
Actually, if you want a perfect example of that last one, if you've ever eaten a seedless watermelon at a summer barbecue, you literally have polyploid breeding to thank for it. By increasing the chromosome sets to create what scientists call a triploid crop, agricultural researchers figured out how to grow these massive, super sweet fruits that just don't produce those annoying hard black seeds. It's this incredible piece of traditional genetic manipulation sitting right there in your fruit bowl.
But as powerful as those traditional methods are, they have one pretty fatal flaw. They take a massive amount of time, sometimes decades. So what happens when traditional breeding just isn't fast enough to beat the rapid pace of climate change? What are we supposed to do when a highly adaptable new pest or a sudden, brutal megadrought threatens our food supply right now?
Section 3. The Biotech Revolution.
That urgent need for speed brings us to a whole new era. We're shifting away from the genetic lottery of traditional breeding and moving toward the absolute precision of a biological word processor. If you look at the comparison, traditional breeding relies heavily on whole plant selection. You cross two plants, plant the seed, grow it, and then wait months or even years just to see if it actually looks the way you want. But modern biotech? It completely flips the script. By using DNA markers, which is known as marker-assisted selection or MAS, and genomic selection, scientists don't have to wait for the plant to grow up. They can look directly at a tiny seedling's DNA and know immediately, right then and there, if it carries the complex traits for things like drought tolerance or high yield. It makes the entire breeding process exponentially faster and way more accurate.
And zooming in on that molecular level paved the way for genetic engineering, or GM technology, where we can introduce specific highly beneficial genes directly into a crop. A couple of classic examples here. First, Bt crops. These actually contain a specific gene from the bacteria Bacillus thuringiensis, which gives the plant built-in insect resistance. That means farmers don't have to spray nearly as many chemical pesticides. Another massive breakthrough is golden rice. This was developed specifically to have increased provitamin A content, and it's designed to combat severe malnutrition and prevent childhood blindness in developing nations.
Now, obviously, this technology doesn't exist in a vacuum, and it's super important we look at the full picture here objectively. The advantages of GM tech are undeniable. Vastly improved productivity, the ability to dial in very specific survival traits, and, as we mentioned, a major reduction in pesticide use, which is a huge win for the environment. But the limitations are equally real. The research costs are absolutely astronomical. Navigating the regulatory challenges can take years. Plus, there are very valid public concerns about long-term impacts and the risk of shrinking our global genetic diversity if we rely too heavily on just a few patented corporate seedlines. It is a really complex balancing act between incredible scientific potential and necessary societal caution. And that complex balance brilliantly illustrates why the next leap forward is getting everybody so excited. Genome editing. Specifically, CRISPR technology. Unlike older GM tech that often brings in foreign genes from other species, CRISPR acts much more like a pair of hyper-precise genetic scissors. The steps are beautifully straightforward. Step 1. You identify the exact target DNA sequence responsible for a trait, say a plant's susceptibility to a certain fungus. Step 2. You utilize the CRISPR-Cas system to navigate right to that exact spot. Step 3. You precisely modify or SNP that specific part of the plant's own genome. And Step 4. You yield an improved, inherently stress-tolerant crop. No foreign DNA required. It's fast, it's incredibly accurate, and it is absolutely a game-changer for yield improvement.
But here's the catch. Editing the genes is really only half the battle. We still have to grow these crops efficiently. To do that, modern agricultural science uses supplementary techniques to seriously accelerate growth and nutrition. For instance, plant tissue culture allows for the rapid multiplication of completely disease-free plants right inside sterile labs. Then there's speed breeding, which is exactly what it sounds like. Scientists use perfectly controlled light and temperature environments to basically trick the plants and churn out multiple generations in a single year. And finally, biofortification. This is where we directly improve the nutritional content of our staple crops. Think iron-rich crops or zinc-enhanced wheat. So when that food finally hits our plates, it is every bit as nourishing as it is resilient.
Section 4. The future of food. All right, let's head out to the bleeding edge. Where is all this cutting-edge technology taking the future of sustainable farming? Well, the future of crop improvement is entirely data-driven. We're looking at AI-assisted breeding and digital agriculture, where artificial intelligence actually predicts the absolute best plant combinations before a single seed is even planted in the dirt. We're utilizing bioinformatics for massive genome analysis and gene prediction, and we're diving deep into omics technologies, like genomics, to study the DNA itself, and transcriptomics to see how those genes actually express themselves when the plant is under stress. All of this high-tech data merging serves one ultimate goal: creating climate-smart crops that deliver maximum nutrition while requiring significantly less water and fertilizer. When you take a step back and look at the evolutionary journey of our food in a single snapshot, the progression is staggering. We started out simply selecting superior plants and combining traits through hybridization, right? Then we moved to forcing genetic variation with mutation breeding. From there, we unlocked the DNA code, which gave us marker-assisted selection, precise CRISPR genome modification, and targeted biofortification. It's like building a giant puzzle. Every single technique builds perfectly on the knowledge of the last, constantly refining our ability to feed a rapidly growing world.
Which leaves us with this final thought. Crop improvement really is an unbroken chain of human ingenuity. It stretches all the way from the earliest farmers saving seeds in a field to modern geneticists programming DNA on a computer screen. But as we accelerate into this new era defined by rapid climate change, what will the delicate balance between natural genetics and human intervention actually look like on your future plate? The science is definitely ready to adapt, but the choices we make today about how we use these technologies, well, that's what will ultimately define the future of our food supply. Thanks so much for joining me for this explainer, and hey, keep asking questions about the science behind your next meal.